So students, today we will do the development of the human placenta. So the essential functional element of the placenta is the chorionic villus, which contains the fetal blood. Outside is the maternal blood, and exchange of material takes place between fetal blood and maternal blood. The maternal component is the heme, while the fetal component is the chorion. So the human placenta is called haemo-choreal. So exchange of material takes place between the heme and the chorionic villi, the fetal blood present in the chorionic villi. So how the chorionic villi are formed? We have done that the chorion is formed from the trophoblast and the underlying extraembryonic mesoderm. Now this trophoblast, it multiplies into two layers, the basal cellular layer called cytotrophoblast, and then there is a syncytial trophoblast. How this syncytial trophoblast is formed? When the cells of trophoblast multiply, they multiply so fast that they lose their cell walls, and a syncytium is formed which keeps on invading the decidua. So trophoblast forms cytotrophoblast, which is the cellular layer, and the syncytial trophoblast, which is invading the decidua. So then small cavities or lacunae appear in the syncytial trophoblast. They increase in size, and these are separated by partitions of syncytium, which are now called trabeculae. So we have the trabeculae shown in black here, and the lacunar spaces shown in white here. So as the endometrium is eroded, the blood vessels in it contain blood and they open up, and now blood fills the lacunar spaces. So you can make out the blood is filling the lacunar spaces. So this is again the same image. Now if I take a section here, students, I will have in the central part syncytial trophoblast surrounded by maternal blood. So here you can make out in this section islands of syncytial trophoblast lying in maternal blood. So if I take a section around, from this point to this point, in the center I will have the syncytial trophoblast, on the sides I will have the maternal blood. So how is the primary villus formed? It is formed when cytotrophoblast cells multiply into each trabeculus. So by day 13 of life, what happens is, by day 13 after fertilization, the cytotrophoblast cells multiply into each trabeculus, and a primary villus is formed, which has got a core of cytotrophoblast surrounded by syncytial trophoblast. So if I take a section from this point to this point, there will be a core of cytotrophoblast in the center surrounded by syncytial trophoblast on the sides, and then there will be maternal blood. So this is the structure of a primary villus. Now how secondary villus is formed? When extraembryonic mesoderm now invades into the primary villus. So extraembryonic mesoderm has invaded the primary villus. Now if I take a section from this point to this point, there will be a core of extraembryonic mesoderm surrounded by cytotrophoblast, surrounded by syncytial trophoblast, lying in maternal blood. So core of extraembryonic mesoderm surrounded by cytotrophoblast, surrounded by syncytial trophoblast, lying in maternal blood. So this is the structure of a secondary villus. Primary and secondary villi are formed by day 13 after fertilization, around about the same time. So how is the tertiary villus formed? This is formed by day 16 after fertilization, when blood vessels can be seen in the mesoderm. So fetal blood vessels are seen here now, and if I take a section now, I will have a central core of mesoderm containing the fetal blood vessels, surrounded by cytotrophoblast, surrounded by syncytial trophoblast, and these tertiary villi are lying in the maternal blood. So from this point to this point, if I take a section, the core of mesoderm surrounded by containing the fetal blood vessels, surrounded by cytotrophoblast, surrounded by syncytial trophoblast, lying in maternal blood. So this is the structure of a tertiary villus. Then anchoring villus is formed, and how this is formed? When the cytotrophoblast forms a shell, and this cuts off the syncytium from the decidua. So here we can make out a shell which is formed, and now we have an anchoring villus, which on one side is anchored to the extraembryonic villus, extraembryonic mesoderm, and on the other side it is anchored to the cytotrophablastic shell. So the villi are now attached on fetal side to extraembryonic mesoderm, and on maternal side to the shell. So this takes place because cytotrophoblast cells cut off syncytium from the decidua, forming a cytotrophablastic shell. So now we have an anchoring villus, which is anchored on one side to extraembryonic mesoderm and on the other side to the cytotrophablastic shell. And anchoring villus consists of a stem which further divides into branches called rami-chori, which further divide into smaller branches called remuli-chori. So they can be appreciated here. the stem dividing into the branches and further branching is taking place. So then a fetal cotyledon is formed. So once anchoring villus is formed, one anchoring villus and its branches, they make up a fetal cotyledon. A fully formed placenta has 60 to 100 fetal cotyledons. So you can appreciate the complexity now, how from primary villus, secondary villus was formed, tertiary villus was formed, anchoring villus is formed, and one anchoring villus and its branches make up a fetal cotyledon. So a fully formed placenta is disc shaped and has got 60 to 100 such fetal cotyledons. If you look at the placenta from the maternal aspect, if you look at it grossly, it is made up of number of lobes. Each lobe is called maternal cotyledon. So there are 15 to 20 maternal cotyledons, and each lobe or maternal cotyledon consists of a number of anchoring villi and their branches. So this one can grossly make out if we look at the placenta from the maternal aspect, the 15 to 20 maternal cotyledons. So if we look at the structure of the tertiary villi, we can make out the components of the placental barrier. What is the placental barrier? Tissues intervening between fetal blood in chorionic villi and maternal blood in intervillous space. So let us see the components of maternal barrier. The basement membrane of the fetal blood vessels. Then we have the extraembryonic mesoderm, then we have the cytotrophoblast, then we have the syncytiotrophoblasts. So exchange of material takes place between fetal blood and maternal blood across the placental barrier. So what are the functions of the placenta? This placental barrier which was formed, it keeps the two streams, the maternal and fetal circulations, separate, preventing antigenic reactions. The placenta is formed essentially to provide nutrition to the fetus, and it eliminates waste products from the fetus into the maternal circulation. The IgG antibodies from maternal circulation, they cross the placenta and provide immunity against some infections like diphtheria and measles. But some bacteria and most viruses can cross the placental barrier. Drugs can cross the placental barrier, causing congenital malformations. So a pregnant lady has to be careful about the medicines she takes, and she should not take any medicine unless the doctor allows her to take those medicines. Maternal hormones do not cross the placenta, but synthetic progesterones and estrogens, they can cross the placenta and cause carcinoma in the fetus in later life. So one has to be very careful. So functions of placenta, it keeps the two streams separate. At the same time, by keeping the two streams separate, it forms a barrier. It allows IgG antibodies to cross through. Some viruses, most viruses can cross through the placental barrier. Drugs can cross through the placental barrier. So when you talk about functions of placenta, it is the selective barrier which is formed, that is important, and then providing nutrition to the fetus and elimination of waste material from the fetus, that is important. Then the placenta secretes hormones, progesterone. It maintains pregnancy after the 4th month when the corpus luteum is not there. So till the 4th month, the corpus luteum keeps functioning due to HCG secreted by the placenta, human chorionic gonadotrophin. And after the 4th month, then corpus luteum is not needed, and progesterone is secreted by placenta to maintain the pregnancy. The estrogens secreted by placenta, they stimulate the maternal uterine growth and mammary gland development. Somatomamotrophin secreted by placenta has got an anti-insulin effect on the mother. What is the meaning of that? That it breaks down carbohydrates into glucose, it breaks down lipids into fatty acids, it breaks down proteins into amino acids. So these simpler forms are available in circulation to be consumed by the fetal circulation. So it increases the availability of amino acids, fatty acids, and glucose in the maternal circulation. Somatomamotrophin secreted by the placenta. So this brings us to the end of the class. The primary villi are seen by day 13. Secondary, tertiary villi by day 16. A discrete placenta, disc shaped, is formed by the 4th month. And I would like to show you a poem which I have written for the placenta as an ode to the placenta. Oh placenta, thanks my dear placenta. So my gratitude. My nourishment was always your agenda. So function of placenta is providing nutrition. From progesterone to waste recycling. So elimination of waste, and it secretes progesterone for maintenance of pregnancy. You were always superb and unrelenting. You were my pillow. So the placenta is the pillow in the coziest womb bed. You sneaked out quickly yet were scrutinized. So when the placenta comes out, one looks for if there is there any, after the birth of the baby, the placenta is taken out and the doctor looks at it to see if there are any congenital abnormalities in the placenta. So you sneaked out quietly, still were scrutinized. My selective barrier, so there the placental barrier is selective. My stoic comrade, placenta assists the fetus. Without you, I would never be me. So students, I thank you for your time, and the next time we meet we will do the abnormalities in the placenta. What are the possible abnormalities in the placenta? So if you need notes on this topic, you can inbox me at 9815542792. So till the next time we meet, it's bye from my side. Thank you.